Today we are going to learn about the cell cycle. So the outline I have formulated here. So first we will look about the basic about the cell and cell discovery. Secondly, we will look about the cell cycle, and thirdly we will see the cell cycle specific anti-cancer drugs. So first that's the basic about the cell. Some MCQs I will ask to you. Who discovered the cell? Who discovered the cell? Robert Hooke. Okay. Robert Hooke. When he discovered? He discovered it in 1665. See, with Robert Hooke, he discovered the dead cells, not the living cells. He discovered dead cells. Then the second point is that who discovered the living cells? Anyone knows? Anton von Leeuwenhoek. Anton von Leeuwenhoek. He discovered the living cells, and he discovered it in 1674. 1674. See, this person he is also referred as father of bacteriology. Father of He's the person who discovered the living cells. Then third point is who gave this cell theory? Cell theory. What is cell theory? Cell theory states that every living organism is made up of cells. That every living organism is made up of cells. And who gave the cell theory? Schwann and Shield. Schwann and Shield. They proposed the cell theory in they proposed the cell theory. Okay. Then after its cell theory, that's cell cycle. Cell cycle. Who gave cell cycle? Who discovered the cell cycle? Or we can say who described the cell cycle? See, Prevost and Dumas. Prevost and Dumas, they discovered cell theory. They discovered the cell cycle in 1824 while studying the cleavage of frog zygote. So cell cycle, Prevost and Dumas, they proposed the cell cycle while studying on the cleavage of zygote on frog. But Walter Fleming, here I will write Walter Fleming. Walter Fleming, he was the first person to draw the different stages of cell cycle. Different stages of cell cycle. So these are some important MCQs. First, robots. This all everyone knows that discovery of cell, it was done by Robert Hooke, but he discovered the dead cells. Then the second important point is living cells. Living cells were discovered by Anton von Leeuwenhoek, and then third, that cell theory, it was given by Schwann and Shield. And fourth important point is that cell cycle, the cell cycle, it was described by Prevost and Dumas in 1824, but Walter Fleming, he was the first person to describe the different stages of cell cycle. Now we will look about the cell cycle. See, what is cell cycle actually? See, cell cycle, it's a series of events. It's a series of events that cell grow and divide. See, as we know that every living organism is made up of cells, okay, and cell, they have to divide it. See, for example, an adult human, it contains nearly about approximate 100 trillion cells. 100 trillion cells. But from where it started? It starts from a single cell, that's called a zygote. So from a single cell, that's zygote, to nearly about 100 trillion cells. So how this comes? This comes from cell cycle, that's cell growth and divide. See, we know that the cell having a property of cells is having a property of division and growth. I will tell you one question. Which cell do not have replication capacity? Which cell do not have replication capacity? After it, when it is normal in the human body, then it does not divide. See, neuron cell is only neuron cell, because that's why, see, whenever there is a neuronal damage, it's a permanent damage. There is not cure. Okay, neuronal damage. See, neuron cell, it does not contain one of the important organelle that is very important for cell cycle, that's centrioles. Neuron cell lacks the centrioles, and this centriole, it's one of the important organelle for the cell cycle. So this is another important point. So now we will go to cell cycle. See, cell cycle, it consists of first, you know that G1 phase, different phases. That's first one is G1 phase. Second is S phase. Third one is G2 phase. And fourth one is M phase. Okay. So G1 phase, actually what is the important events which are going in this given phase is that there is cell growth. For example, see, I will lastly I will put the diagram, then you can understand it. Here you only just concentrate about the key activities or functions. In the G1 phase, there is cell growth is there. There is cell growth. When the cell wants to divide, the first phase is that this cell it will grow, and it will prepare for DNA synthesis. Okay, so this is the first phase, the G1 phase. Okay, so in this phase, the cell cycle the cell which wants to divide, okay, it will undergo the growth, and it will its size will get enlarged, okay, for the preparation of DNA synthesis. So this is the first event in G1 phase. In the S phase, there is the DNA synthesis, or you can you can remember that as DNA replication. In the S phase, there is DNA replication. Okay. In G2 phase, there is division of nucleus. Okay, in G2 phase, there is cell prepares for mitosis. And here organelles, proteins and organelles are formed. Okay, and then the M phase. In the M phase, there is division of nucleus and cytoplasm. That is division of nucleus and cytoplasm. See, these are 4 important phases of cell cycle. First one is G phase. In G phase, cell tends to grow and makes its size enlarge and prepares for DNA synthesis. In the S phase of cell cycle, there is cell prepares for, or we can say there is DNA replication is going on. See, and DNA it will form. If this is a single copy of DNA, it will form double copy of the DNA. That's the DNA replication. Okay, and in G2 phase, there is cell prepares for mitosis, and there is formation of organelles and proteins. In M phase, there is division of nucleus and cytoplasm. See, these are very important pointers in every MCQ. In every exam, they can ask in terms of MCQ. For example, DNA replication is carried in which phase of cell cycle? Then there will be option G1 phase, S phase, G2 phase, M phase. You have to mark the S phase. So S phase, there is DNA replication, and G2 phase, and another important, see, the question might come like formation of organelles and protein. In which stage of cell cycle there is formation of organelles and proteins? You have to mark G2 phase. And in M phase, there is a division of nucleus and cytoplasm. Okay, so see, and M phase is further divided into following. M phase, okay, that's in M phase what we have, that cell division of nucleus and chromosome separation. Chromosome separation. Okay, so in M phase it's further divided into, that's first one is prophase. Prophase. Second one is prometaphase. Prometaphase. Third one is metaphase. And fourth one is anaphase. Anaphase, and last one is telophase. Telophase. See, in prophase, what's happening here? What's the key event? There is chromosome condensation is there. In prophase, there is chromosome condenses. Okay, and it becomes visible, as sister chromatid. So in prophase, the chromosome they condense and becomes visible. So this question is asking like, in which phase the chromosome becomes visible? So you have to mark prophase. So in the prophase, chromosome becomes visible, and there is a formation of sister chromatids. Then prometaphase. In prometaphase, there is nuclear envelope development. Nuclear envelope. Disintegrate. See, here, for example, this is a cell, okay? This is a cell, okay, and this is a nucleus. See, first in the prophase, what will happen? The chromosome, they will become visible and apply as a chromatid system, and visible as a sister chromatid, like this, okay? They will become visible, okay? In prophase, the chromosomes become visible and appear as a sister chromatid, like this. In the then in prometaphase, this nuclear envelope, see, this is a nucleus and this is a nuclear membrane. It will disintegrate. And these chromosomes, they will come into cytoplasm, okay? So in prometaphase, there is nuclear envelope, disintegration is there, okay? Okay. In metaphase, see, the chromosome align at the center of the cells. In the metaphase, chromosomes, they will align at the center of cell. See, here I will write. See, this was the previous event. In the next event, what will happen? This nuclear envelope, it will break in the prometaphase, and now in metaphase, these chromosomes, they will align in the center. Chromosome align at the center of the cell. And then in anaphase, centromere splits. Separating the sister chromatid. So they will separating the sister chromatid. And in telophase, see in the telophase what happened? In the telophase what happened? There will be a growth of nucleus inside. See, this is a cell, this is a cell, and there will be formation of again nuclear membrane, but there will be formation of two nuclear membrane like this, okay, containing chromosomes. Okay, so this phase it's carried in telophase. So in telophase what's happening? In telophase, yeah, first before that, in telophase, first the chromatid here, they will reach to opposite ends like this. They will reach to opposite poles. They will reach to opposite poles, and then subsequently there will be the formation of nuclear membrane around the chromatids, okay. Then the final stage, that cytokinesis is called. See, then this it will form into the two cells. This is a nucleus containing chromosomes, and this is this containing chromosomes. Then the cell further might go under cell division, or it will, see, if it further goes under cell division, this will be carried through G1 phase. If it remains stagnant, then it's the G0 phase. Okay, so these are important events. I will draw the diagram, then you can understand. Okay, this is a cell. So when it undergoes cell cycle G1 phase, first important point is that here this cell will grow in size. Size will get increased, okay, and it will synthesize proteins and enzymes for DNA synthesis. So it will get involved in the size, and it will synthesize proteins and enzymes for DNA. For example, these are these are enzymes and proteins. In this stage there is no enzyme, there is no protein, but in this stage there is enzyme and protein, okay, for DNA synthesis. So this is the G1 phase. In the next phase, that's S phase, there is DNA replication is there, okay. See here, only one DNA is there. In this there is double DNA. So there's a DNA replication. In another phase, that's this is the S phase. In another phase, that's called a G2 phase. There is formation of cell prepares for mitosis, okay. Cell prepares for mitosis, and there is formation of some organelles. Like centromere. So this is the G2 phase. And then M phase. In M phase, what will happen? See, M phase is split in 5 phases. Then cell division occurs in the M phase. How it will occur? So there will be, we have in M phase, the first phase is what's that first phase in M phase? Tell Hemant. In M phase, see, first is prophase. See, prophase. See, there is formation of spindle. Centrosome, see, this is a centrosome, and in the centrosome there is formation of like spindle formation, okay. So in prophase there is formation of spindle. Okay, this is the prophase. In next phase, that is 1 minute. See, this is a prophase. In then sex, that's called that is prometaphase. Prometaphase. This nuclear envelope, it will disintegrate. So nuclear envelope, it will disintegrate, and the chromatid, it will come in contact with cytoplasm. This is the nuclear membrane disintegration. Okay, in the next stage, this is called this is prometaphase. Prometaphase. In next phase of cell cycle, that's metaphase. Okay, metaphase. Chromosome, they will align at the meta plate, like they will align at the center. Okay, and in anaphase, here I will write, in the anaphase, sister chromatids are separated and pulled apart, like sister chromatid. These, see, like here it was like this, and here sister chromatids are separated and aligned. In the telophase, these chromosomes, it will be pulled on opposite poles, and there will be the formation of nuclear envelope. Then in cytokines, in the last, that's cytokinesis, there will be formation of two daughter cells, and then the cell either go under cell cycle or it will remain at a stagnant. Is called genome phase. So we have some drugs. After this I will finish this. See, first that's if we look the classification of anti-cancer drugs, that we have cell cycle non-specific, and then we have cell cycle specific drugs. Cell cycle non-specific, you know that the example is the cytotoxic drugs. That's cisplatin. Okay, cisplatin, oxaliplatin, all these nitrogen mustard drugs. Okay, these are cell cycle non-specific. And in cell cycle specific, we have specific. We have specific, which will act on the specific phase of cell cycle. For example, in the G1 phase, which drugs will act? Hormonal drugs will act. And enzymatic drugs will act. See, hormonal drugs, example, what's the example of hormonal drugs? Okay, hormonal drugs, you know. See, and enzymatic drugs, that's L-asparaginase. Okay, hormonal drugs, we have a huge classification, like anti-androgenous, all these things. Okay, then they will act in the G1 phase. Then in S phase, see, this is the most important, anti-metabolite drugs, which will act on the S phase. For example, pyrimidine antagonists, purine and pyrimidine. Okay, like mercaptopurine, and then we have the examples. Okay, so they will act in the S phase of cell cycles. That's anti-metabolite drugs. That's folate antagonist, methotrexate, purine analogous, cytarabine, purine analogous, cytarabine, all these things, they will act. See, you just keep the name, that's anti-metabolite. They will act on the S phase of the cell cycle. In the M phase, see, very important drug, that's vinca alkaloids will act on the M phase. That's vincristine and vinblastine. They will act on the M phase. Okay, in G2 phase, miscellaneous drugs like antibiotics, bleomycin. bleomycin, they will act in G2 phase. So this was brief about cell cycle and cell cycle specific anti-cancer drugs.